Here, let us perform an activity to check the presence of starch in leaves. Click each tab to know more. Let us take a soft thin leaf from a potted plant. Boil the leaf in water for 5 minutes to soften it. Place the leaf in a test tube containing alcohol. Take a test tube with methylated spirit and place the leaf in it. Now, place the test tube in a beaker containing water. Gently heat the test tube in a beaker containing water until the leaf becomes pale white. Note, this happens because the chlorophyll gets removed from the leaf. Now, observe the leaf and wash it with water. Next, place the leaf on a petri dish. Add few drops of iodine solution to it and observe the changes in the leaf. It is observed that the parts of the leaf turns into blue-black color, indicating the presence of starch. From this activity, we learn that starch is present in the leaves even in the absence of chlorophyll. Joseph Priestley, who discovered oxygen in 1774, performed a series of experiments to reveal the essential role of air in the growth of green plants. He observed that when a burning candle is enclosed in a bell jar, the flame of the candle is gradually put off. In the same way, a mouse also suffocates in a closed space, such as in an enclosed bell jar, and dies. From these observations, Priestley concluded that a burning candle and an animal change the composition of the air. He reasoned that something was necessary in the air to keep the flame burning and to make the mouse stay alive. Priestley then placed a mint plant inside the same bell jars, and he found that the mouse stayed alive and the candle, when lighted from outside, continued burning in the presence of the mint plant. Priestley hypothesized that the plants produced a gas, oxygen, that supported the burning of the candle and also allowed the mouse to survive respiration. The production of the gas takes place due to a massive exchange of gases occurring through a particular section of the leaves called stomata, and also through loose tissues on stems, roots, etc. Let us discuss in brief about how carbon dioxide is useful for photosynthesis. Here, let us perform an activity to learn about the necessity of carbon dioxide for photosynthesis. Click each tab to know more. In order to start the experiment, we need a destarched plant. For this reason, place a plant in the dark for 3 days in order to free the leaves from the starch. Remember the process of destarching the leaves as performed in activity 1, presence of starch in leaves. Insert a splitted cork in the mouth of the bottle. Place half of the leaf of destarched plant into the bottle having potassium hydroxide pellets or potassium hydroxide solution. Leave the plant under sunlight for few hours. After this, test the part of the leaf inside the bottle with the part of the leaf exposed to sunlight by pouring iodine solution on it. Observe the changes in the leaf. It was observed that half of the leaf was exposed to the sunlight. Atmospheric air becomes bluish black, and the leaf which was present inside the bottle containing potassium hydroxide does not become bluish black because potassium hydroxide absorbs the carbon dioxide present in the bottle. This indicates that carbon dioxide is necessary for the process of photosynthesis. From this experiment, it is clear that carbon dioxide is essential for photosynthesis. Now, let us do an activity to learn about the production of oxygen during photosynthesis in the presence of sunlight. Here, let us perform an activity to understand how oxygen is produced during photosynthesis in the presence of sunlight. Click each tab to know more. Consider the apparatus shown in the image. Take a beaker and fill it with pond water. Place water plant like hydrilla or elodie inside the beaker and cover it by a short-stemmed funnel. Take a test tube and invert it over the stem of the funnel. Ensure that the level of water in the beaker must be above the level of the end of inverted funnel. Place the entire assembly of apparatus in the sunlight for at least 3 days. It is observed that in place of water, air is filled in it. This air is actually a gas and is collected in the test tube. Test the gas collected in the test tube by inserting a glowing incense stick, which would burst into flames. This indicates the presence of oxygen. The air bubbles formed in the test tube is of oxygen gas. Thus, it is clear from this activity that oxygen is produced during the process of photosynthesis in the presence of sunlight. Here, let us perform an activity to learn about the need of sunlight in the formation of starch. Click each tab to know more. Take a plant having destarched leaves, that is, a plant which is placed in the dark for 3 days. Remember the process of destarching the leaves as performed in activity 1, presence of starch in leaves. Wrap one of its leaves with black paper on which a design is cut. Fix the black paper on the leaf with the help of clips such that light should not enter the dark part. Now, place this plant in the sunlight for few hours. Later, test the leaf that is covered by black paper for the presence of starch by pouring iodine solution on the leaf. Observe the changes in the leaf. It is observed that only the parts of the leaf which are exposed to the light through cut-out design turn to blue-black, indicating the presence of starch. From the experiment, we learned that starch is formed only at the parts where sunlight falls. In this way, it is clear that sunlight is compulsory for the formation of. Different organisms have different strategies for consuming the food. Few organisms break down the food material outside the body and then digest it. Examples: bread molds, yeast, mushrooms, etc. Some other organisms like amoeba take in whole food material inside their bodies and then break down into small pieces. This depends upon the digestive system of each organism. Some other organisms like cuscuta, lice, leeches and tapeworms take the nutrition from plants and animals without killing them. Let us learn how organisms obtain their nutrition. The digestive system is different in different organisms. For example, in a single-celled organisms like amoeba, the food may be consumed from the entire surface, but as the size of the organism increases, the parts become specialized and perform different functions. The nutrition in amoeba is shown on screen. The amoeba takes in food by using pseudopodia as shown in image. This pseudopodia looks like temporary finger extensions of the cell surface which fuses over the food particle, forming food vacuole. Inside the food vacuole, the complex substances of food particles are crushed down with the help of enzymes into simpler ones and then diffuse into the cytoplasm. The undigested food particles are moved down to the surface of the cell and are thrown out. In other unicellular organisms like Paramecium, the cell has a definite shape and the food is taken in from a particular place, and this food is moved to the place by the movement of cilia that covers the entire surface of the cell, where the food gets swallowed. Let us learn in detail about parasitic nutrition in Cuscuta. A particular species in Cuscuta is dodder. It does not contain any leaves. It is a twining, parasitic plant that belongs to Morning Splendor family. These species generally contain 170 twining species and are broadly distributed throughout the moderate and tropical regions of the world. The dodder species does not contain chlorophyll. It absorbs food through haustoria, which are root-like organs that penetrate towards the tissue of a host plant and even may kill it. But it has been found that Cuscuta reflexa contains a very small amount of chlorophyll. The thin, string-like stems of the dodder may be of different colors like yellow, orange, pink or brown. The dodder's flowers are of tiny yellow or white bell-like, lobed corollas, united petals. Let us now learn about the formation of dodder. The dodder seed grows slowly and forms an anchoring root, and grows in a spiral fashion and reaches a host plant. It then waves around the stem of the host plant and throws out haustoria, which diffuses it. The water for the species is drawn through the haustoria of the corresponding host plant's stem and xylem, and the nutrients are drawn from its phloem. In the meantime, the root decays after stem contact has been made with a host plant. As the dodder grows on, it sends out new haustoria and sets strongly on the host plant. Suppose, after growing a few spirals, if the host decays, then the dodder finds another way and continues to wave and branch until it looks like or becomes a fine, densely tangled web of thin stems enveloping the host plant. Now, let us learn about nutrition in human beings. When compared to the digestive system of other organisms, human digestive system is very complex, where different parts perform different functions by using various digestive juices and enzymes. Image on the screen shows the alimentary canal of man. The alimentary canal is a long tube extending from the mouth to the anus, as indicated in the image. This tube has various regions which perform different functions. Generally, every one of us get doubt about what happens to the food once it enters into our body and how it gets digested in the alimentary canal. Let us learn more. Usually, everything we eat passes through the same digestive area. The process of taking food into the body is called ingestion. Naturally, the food has to be processed to generate simpler particles from the complex substances, because the particles must be small enough to be picked up by our body. The texture also needs to be easily absorbed. Let us discuss in detail about the steps involved in the passage of food through alimentary canal or gut. The food we eat is chewed by our teeth in the mouth. This chewed food is mixed with saliva to make it wet and slippery. The saliva is produced by 3 pairs of salivary glands that are situated at the side of the jaw and below the tongue. The tongue helps in mixing the food and forwarding it to the next part, and the lower jaw also helps in the whole process. These parts help in the smooth passage of food through our alimentary canal to the stomach. Saliva contains an enzyme amylase named as ptyalin, which helps to break down the complex substances of food into simpler ones so that they can be used by the body with the help of enzymes. This process is termed as digestion. Drag and drop puzzle. From the image on screen, identify the parts of the digestive system from mouth to anus. The first one is done for you. Knowledge check. Attempt the following. Here, let us perform an activity where the acidic and basic nature of food materials are tested using litmus paper. Click each tab to know more. Take 2 litmus papers, one red and the other blue. Touch the 2 litmus papers one by one to your tongue before taking food into the mouth. Observe whether the litmus papers change the color. It is observed that the color of the 2 litmus papers remain unchanged. Now, let us do an activity to learn about the nature of our saliva after chewing the food. Repeat the same test after chewing the food, orange, and swallowing it. Now observe the change in color of the 2 litmus papers. It is observed that the color of blue litmus paper turns into red and the color of the red litmus paper remains unchanged. Generally, in neutral state, color of blue litmus and red litmus paper remains unchanged. In acidic state, color of blue litmus paper changes into red and red litmus paper remains unchanged. In base state, color of blue litmus paper remains unchanged and red litmus paper turns into blue. Thus, from this experiment, we have understood that before chewing the food, neither the red litmus nor the blue litmus paper has changed the colors. This is because our tongue is slightly alkaline in nature, neutral. After chewing the acidic food orange, then the color of blue litmus paper turned into red and red litmus paper remained unchanged. After chewing the base food, say tomatoes, the color of blue litmus remains unchanged and the color of red litmus paper turns into blue. Let us now learn about the movement of food from esophagus to stomach. It is technically called peristaltic movement. The soft food that is mixed with saliva passes through esophagus or food pipe to the stomach by a wave-like movement called peristaltic movement. In the stomach, the food gets mixed with gastric juice and HCl, which is in a semi-solid condition. The digestion of food goes on as most proteins are broken down into smaller molecules with the help of enzyme pepsin acting on them. Chyme is the process in which the proteins and carbohydrates of the soft, slimy substance of food are broken down. The food now passes from the stomach to the small intestine, which contains ring-like muscles called as sphincters. These sphincters are responsible for opening the valve so that an only small quantity of the food material enters into the small intestine at a time. Let us learn about the small intestine. The small intestine is the longest part of the alimentary canal. Here, the complete digestion of carbohydrates, proteins and fats takes place. Small intestine receives the secretion of liver and pancreas for the purpose of digestion. The process of digesting the fats by converting them into small bubble-like substances with the help of the bile juice that is secreted from liver is called emulsification. Proteins and lipase for fats are digested with the help of enzymes like trypsin present in pancreatic juice. Walls of the small intestine secrete intestinal juice, which helps in breaking down of small molecules of proteins into further smaller molecules. The same process is repeated for fats. The process of digestion of carbohydrates starts in the mouth. It resumes in the stomach when the food is converted into an alkaline form. Here, the enzymes responsible for the breakdown of carbohydrates become active. The table on the screen gives information about different enzymes, digestive juices, and its functions. Let us learn the function of villi in digestive system. The process of transporting the digestion of products from the intestine into blood through the wall of intestine is called absorption. Internally, the intestinal wall contains a number of fingers-like projections called as villi. The villi increases the surface area for absorption. Blood vessels and lymph vessels are present inside the villi in the form of a network. The digestion of products are first absorbed by the villi and then passes into the blood vessels and lymph vessels. With the help of small intestine, the maximum amount of digested food is sent to various parts of the body through blood. Remaining amount of the food material passes to the large intestine. Large intestine reabsorbs maximum amount of water from the remaining food material. This material is then passed through the anus, which is the last part of the alimentary canal. The process of passing of undigested material from the body through the anus is called defecation. Food that comes out from the anus still contains significant amounts of proteins, fats, and carbohydrates, roughages or fibers of either carbohydrates or proteins. Knowledge check. Attempt the following questions to check your understanding. The flowchart of human digestive system is shown on the screen. Excess or improper intake of food cause health issues with the alimentary canal. Let us learn about all such aspects associated with the alimentary canal. The human alimentary canal exhibits good functionality, though we have some indigestible food like junk food, etc. But at times, the alimentary canal cannot take some food, as a result of which we either feel sick or have indigestion. The human body gets rid of the unwanted and harmful substances present in the stomach through vomiting. When the peristaltic movements of the stomach and esophagus occur in reverse direction, it results in expulsion of the food. Among all the causes of vomiting, the most common cause is overeating, especially the food which has high proportion of fat. Vomiting also occurs when very indigestible and poisonous food is consumed. Bilious or liverish feeling is often due to consumption of rich meals over several days. Liver cannot cope to digest the food which is rich in fats. As a result, we suffer from feeling of nausea and headache sometimes. Indigestion is a general term used when there is difficulty in digesting food. Healthy people do not suffer from indigestion if they follow below mentioned diet. Having simple and well-balanced food, eating them in a leisurely manner, chewing the food thoroughly, avoiding strenuous exercises soon after having food, drinking plenty of water and having regular bowel movements. Humans should learn a lot from the animals which have a good sleep after having food. Indigestion caused by stomach and duodenal ulcers is very severe. This problem is often observed in people who are worried and hurried. So, ulcers are mostly observed in people habituated to have hurried meals and rush from one work to other and don't have proper sleep. Example, doctors, schoolmasters, members of parliament, stockbrokers, and business executives. The people who maintain a proper diet, have good sleep, and don't tense a lot seldom get ulcers. In order to stay healthy, the bowels ought to be emptied regularly. If the residues of food are present in the colon for much longer time, the bacteria get sufficient time to produce harmful substances which might be absorbed by blood and cause diseases like cancer, etc. Having plenty of roughage, fiber in the diet helps a lot in avoiding constipation.